waves get lost in the ocean. 7 billion swimmers, man, I'm going.
So as we've heard, the MSCRE is really about supporting research from all different directions, and one of the activities from the MSCRE is giving travel awards. And so today's final two presenters in this session are Dr. Xin Lin and Meg Denham, and they're both going to be presenting on based on research funding that they've received from the CRE to support innovation and travel awards. So the first presentation is from Dr. Xin Lin, and he's going to tell us about his work understanding early immune changes in MS before treatment starts. Thanks, Xin. Come on up.
Thank you so much for the introduction, Tracy. Hi, everyone. My name is Xin. So I'm one of many researchers that have really greatly benefit from attending events like today. And it's been a real privilege to do MS research with connections to the community of people with lived experience of MS, as well as researchers across different disciplines. And and I've acknowledgement slides towards the end, but I'd like to begin with a thank you to this community who made this work possible. So today, I'd like to tell you a bit about this work got funded by Innovation Award last year. And it's about understanding the early immune changes in MS before treatment starts. So why does this matter? The immune system is the body's natural defense against infections and disease. And in MS, it mistakenly attacks the brain. And changes in the immune system starts early, so before noticeable symptoms or signs on the brain scans appear. And understanding these changes could really help us better detect MS and monitor MS. And ultimately, this will improve care and health outcomes and reduce long-term disability and costs. And this work uses an innovative approach to uncover previously invisible immune activity in MS. So what it's about, it's to detect these, what I like to call immune ID badges in the blood. So our cells show these tiny protein pieces called immunopeptides as ID badges on the surface of the cells for immune system monitoring. So an immune cell can come around and check these ID badges to tell whether it's friend or foe. But in MS, we still do not know which of these immunopeptides appear early in MS and may act as early disease signs. So here we are using a new approach to study these early immune activity in MS. So we've collected and analyzed blood samples from people with MS before they starting treatment. So these are newly diagnosed MS individuals, as well as matched controls, so people without MS. And we measure and analyze these ID badges from their blood, and we collect real-time immune information. And this advanced MS research by having this new tests allowing practical and human-centered MS research looking into active immune responses. So in the past year, after we got this funding, we have done the experiments, and using the latest technology, we could actually see previously invisible immune signals in the blood. So what we saw were thousands of these immune ID badges, mostly derived from humans, but also a couple from the Epstein-Barr viruses that you heard a bit earlier in the talks. And these were directly detected being shown to the immune system. And these signals appeared before disease-modifying treatments. So shown here in the figure is this protein ID badges being shown to the immune cells. So what does this mean? So this is a new way for us to look for early signs of MS in the blood, and it's done with a flexible setup that can continue to grow in terms of examining blood samples from more volunteer participants and support ongoing research into important unanswered questions. And over time, this could help to identify who may need closer monitoring earlier, support more informed treatment decisions, as well as ultimately support improving the quality of life of people living with MS. So in terms of consumer involvement, the MS community has played an important role in identifying neuroprotective and reparative treatments as a top research priority. And this work will contribute to ongoing efforts within the MS CRE objective, as well as the MS research flagship in better understanding the MS biology to accelerate progress towards this identified top priority research area. And we also have established a consumer partnership with Meg Denham, who's in the audience, and will present shortly after me. And she contributed also to our recent grant submission that includes looking at proteins as a key component in this research, and I plan to incorporate further inputs and lived experience perspective from Meg as this project progresses. So these are the project contributors and project funding, mostly supported by the CRE Innovation Award, and other sources of funding also include MS Australia and MRFF. And these are the event contributors and funding. So that's all my slides. Thanks so much.
Hang around, we'll see if I forgot that I'm mic'd up here, but not say anything odd. We do have 5 minutes if people would like to ask Jin any questions before going on. I think you're off the hook, Jin. Thank you for your presentation and congratulations on your award.
Real friends, good friends.